Hey everybody, how are you doing? Today we're going to be talking about our 3D printing project. And to kind of get ready for that, I thought I'd go over a handout that I prepared to sort of talk about length scales. And this is going to go beyond this course's reach for a few minutes, but let's have some fun. Hypothetically, roughly the smallest scale of nature that we can conceive of is something called the Planck length. And what is that? That is just a combination of physical constants, things like Planck's constant, speed of light, etc., that give us a length that we think is sort of the smallest length you might have. It's not necessarily a real thing, but if you look at it, this is 35 decimal places. It's a lot smaller than anything else we know about. Another scale that we sometimes talk about is a zeptometer. And this is, if the atom were the size of a person, a zeptometer would be the size of an atom. And this is 1,000 times smaller than we believe an electron to be. So this is really the limit of actual physical measurements that we have done. 10 to the minus 18, I don't have a good answer. That's AM. I don't recall what that is right this minute. But electrons we know are smaller than this. And how do we know that? Well, we smash them together and we can measure how close we're able to get them. And so we're able to get them at least closer than that, and there's nothing apparently inside of them. You go up 1,000 times and you get up to a proton. You go up 1,000 times more and you get up to the francium atomic radius. So again, we're still talking nuclei of atoms. You go up 100 times from that, and now you're looking at atomic sizes, right? So this is really the range in which we're going to start thinking about our 3D printing project, is somewhere around here, right? We're probably not going to print a lot of things that are 1 angstrom big because that's not that physically interesting. If you think about it, if you make a single carbon-carbon bond, let's say you were going to print something that had a carbon-carbon bond and you were going to print it to scale. And you decided that, okay, this is 1 centimeter long right here. Well, as soon as you're looking at molecules, which are roughly 10 carbon bonds, you're looking at something that's 10 centimeters. Right? And if you don't recall your metric very well, 10 centimeters is about 4 inches. And if you then want to look at 10 molecules, you're now looking at 100 centimeters or a meter in size. And our 3D printers are not going to be able to build something that big. So when we're 3D printing, we need to kind of decide what are roughly the 3 decades that we're going to be thinking about, because our 3D printer is going to be stuck within that. I really want whatever you 3D print to be printed to relative scale. And that doesn't mean that you can't print some cellular process and then take a small part of it and make that part much bigger. But whatever's in the main picture, the main story, all has to be printed to relative size. If you haven't yet, please, pause this video. Everyone go out into the hallway. Well, pause after I'm done talking. But, everyone go out into the hallway over by the MPC, and there you're going to see a 3D printing project that I was pretty happy with. And you're going to see a cell, and it was infected. And they decided, okay, we're going to use actual color printing to print some background. We're then going to print pieces, and then we're going to attach those pieces onto that picture. And then, as you'll notice, they made a number of subunits much larger on the side. And I really want you to do something like that. And I don't know what that thing is going to be. What I know is that you are all collectively more creative than I am. You are all collectively more able to come up with something cool than I could possibly do and assign you. Okay, so let's come back to this list, right? Molecules are roughly nanometer size. 10 times bigger than that, you start to get to proteins, then to organelles, and then to bacteria, right? If we're going to actually say, let's say you were like, you know what? I want to print a bacteria, and I have found somebody in the 3D database that has a bacteria 3D model. Fantastic. I'm going to make that. Well, what are you going to see at that scale, right? Are you going to try to print something 10 times bigger than that, 10 times smaller than that? I would guess that your bacteria is probably the upper end. So now we're printing proteins, organelles, bacteria type size However, too small probably for us to be able to see as molecules. Although maybe, if you look at one of the other 3D projects, people had used little pins with heads on them to try to get that scale. And maybe that's the scale you're going to do. What are you going to print? I really don't know. If you look in the classroom, right, I've got those two posters, and one shows cellular processes, the other shows metabolism. You might go over to that and go, I want to know more about this part. And then someone else is looking at the NIH database. This is a huge brainstorming activity, right? You're going to spend all of class today on Friday thinking about this and just trying to look at the list of ideas that you all have come up with and try to decide what's the right thing. Now, there is also a degree of practicality here, right? The right thing might not be your favorite thing. It might be the thing that you can find 3D models for. So this is what I need you to understand. Anything you find in the protein data bank, which you accessed already for your homework, right? rcsb.org. Anything you can find in the 3D data bank or in the protein data bank, I can print. So you check that out. You find a structure. You say, Dr. Dave, I want to print it. I will make a 3D model of it. I will print it. Now, let's say you're like, but I don't want and generally I print atomic representations. So that we're clear, right above me in this image are a bunch of atomic representations of things. And that's surface representations. That is, we're saying, where is the surface of the electron interaction that occurs on this? That's the sort of structure I can print really easily. Let's say, however, you want to print something with a jelly roll. If you don't know what a jelly roll is, it's a certain kind of protein fold that is used for stabilization. I want to print something with a jelly roll, or I want to print something with really neat beta sheets. I can do those kinds of prints as well, but I just need to know that that's what we're doing and we can make those representations. And sometimes it's useful to see something from its secondary structure and its tertiary structure point of view in proteins. If you don't know what I'm talking about, ask people in the class. Somebody does. Okay, so, once we're at bacteria, 10 times bigger than that is eukaryotes, 10 times bigger than that is sort of your human hair and your width. And we can just keep going, 10, 10, 10. Your pinky, the width of your arm, a 1 meter, a school bus. I don't know why I use that. I guess everybody knows it. I needed something that was about 10 meters long. And we can just sort of keep going, 10 times bigger, 10 times bigger. So Usain Bolt, can run 100 meters in 9.58 seconds. And as it turns out, if you take his speed and divide it by his height, you actually get the same ratio that you do for E. coli and their speed. It's pretty wild. They are the Usain Bolt of the microbial world. It's pretty awesome. So as we just keep going, we keep getting these other distances and other distances. James Webb Telescope, closest distance to Saturn, 1 astronomical unit, on and on and on and on, until suddenly I'm jumping, by powers of 4, powers of 2, powers of 4, right? It's huge. And the observable universe is about 93 billion light years. So all in all, as far as we know, everything in our universe spans 61 decades of those powers. All right, let's talk about some things that you could 3D print. Let's say you want to make a bacteria cell and a yeast cell. And maybe you like the stuff that we already have in the classroom. You're like, look, why reinvent the wheel? Let's print things and add on to something that's been done. I don't mind that. But we have to figure out how many of those things we're going to print, what size they're going to be, where we're going to get those models from. This really is this is what we want to do. What can we find? Okay, that doesn't work. Let's do something else. What can we find? There's a lot of back and forth here, and you have to just be willing to be creative as you try to figure something out, right? Real world projects, you have to be inventive. You have to figure out what you can do, and that's what I want us to be getting from this, right? So if I wanted to build something bacterial, that's roughly what it's going to look like, right? If I wanted to build something to the yeast size, that should be what your 3D model resembles at the end of the day, and that gives you an idea for how many of those components you're going to have to make, right? How are we going to get a structure for the ER? I don't know. That's for you to decide, right? Maybe you're like, you know what? Mammalian cells are bust. Well, there you go. How am I going to do actin filaments, pipe cleaners? I don't know, right? You're going to figure out what this model looks like, and you've already proposed a huge and awesome number of things. So if we take a look here, whoop, maybe I'm in the wrong. Oh, here we go. So these are the ideas that you all have proposed, right? DNA helicase, right? So a large printing of DNA with strands wrapped around it. That could be fun. I don't know. HIV attachment and fusion to the CD4 plus cells. HIV is a really interesting virus. It actually starts out spherical, which is similar to the kinds of viruses that I start studying in my research. And then it has this cleaving that happens, and it suddenly looks more like an egg. And nobody knows why. Nobody has a clue. The plasma membrane of RBC, right? Hemoglobin, when it's bound to a different number of oxygen atoms. None of these are like full things. These are just things that we might print and figure out a way to put them together because we're trying to tell a story. Think about how much you've learned in this class just by looking at things drawn to the correct biological scale, right? That's really shown us a lot. If we are looking at this, we go, wow, I've not seen a cell drawn in that level of detail before. So that's the sort of thing I want you to do and to really emphasize when you make your model is what story can you tell? How can you help someone see the magic of the biological world in a new way? This is really your job. And, if this goes well, if this works out well, this is a story that you can tell going forward. You are in a grad school interview, you're in a med school interview, and they're like, tell me about something in your undergrad that you really enjoyed. Maybe it's this project. Maybe it's the building the kinematic model. Let your passion enter this project, right? Most people are not just STEM people. Most people have a variety of interests. We've got people who do art, people who do music. I don't know how you bring music into this, but maybe you do. Maybe you add it in. Maybe you're really good at sketching and drawing and things like that. Maybe you come up with a storyboard idea, right? Maybe you don't make a giant single scene. Maybe you tell snapshots of a story. Maybe you like cinematography and you've got vision for telling a story in that way. I don't know what that is. You know what that is. And I really invite you to bring this in. I'm offering you a grade for this, right? Let yourself have a good time. Explore, play, enjoy yourself. Okay? All right. Back at it here. Maybe I want to tell stories of these organisms together. I don't know. It really is your project. I want you to enjoy your project. This is an environment that you have the freedom to learn. Okay? All right. Back to some of your ideas. Bacteriophage infection cycle. There's one of those in the hallway. Maybe you can do it better. I don't know. Right? There are so many awesome ideas here. And as we're looking at them, there's so much we could do. Right? A nucleosome complex with histones. That would be amazing. Right? I would love that. These models can be found. We can print these subpieces. Maybe we make a nucleosome complex that can come apart. And maybe we get some magnets and we take the DNA and we have the DNA pieces and they've got magnets on them and we can attach them to the histone proteins and make the full nucleosome complex. And someone can pull it apart and see all the pieces of it. I don't know. Maybe we make some chunks of DNA that are methylized so that we don't want to express them and we show what that does to its structure. Again, I don't know. Or you will figure it out. Right? That's what we're doing all of today on Friday is we are brainstorming and coming up with ideas. Now, let's be clear. This is what happened the last time I taught this course. We had roughly 2 camps that wanted to tell different stories. And they decided they couldn't mix it together and no camp wanted to give up. So they gave 2 different stories. And I think that they were somewhat diminished by that. So get attached to your ideas, but don't be willing to, or don't be afraid to shift, right? Let's work together. And again, it's going to take some leadership, okay? And leadership doesn't mean someone's in charge, right? Because you're all just students, right? But leadership doesn't mean being in charge. Leadership means bringing up voices of other people. Leadership means making sure that everyone gets to speak up and be part of it. And so I encourage you, step up and try to show some leadership and help bring this together. Okay. I love this notion of like the largest and the smallest bacteria. That would be really neat, right? If we can find structures for those, that might be a lot of fun. But again, I don't know what we can find out there. I haven't played with the NIH database in several years. I used to be on it all the time and did all kinds of prints from it, but now I make most of my own models that I want to print, so I'm honestly not as reliant on it. Okay. Something with neurons and neurotransmitters. That's another really fun idea, right? G protein coupled receptor and its subunits interaction. That might be a really neat story, right? But again, that's a story that you're going to tell. Now, once we get an idea of like what we're going to do and we've really kind of storyboarded it out and we found a number of 3D print models that we might use, we're going to start getting a hold of Josh Moon over in the library, and we're going to start sending him some of those models, and we're going to start doing some test prints just to see how they look, right? And here's another fun one too. When you 3D print, you get a whole range of colors. So how can color influence your story, right? How can size influence your story? If you need building materials to do this, I can order those, right? So the model that you see in the hallway of the virus, all that stuff attached to the poster board, there's actually a metal plate behind that and magnets attached to it. So if we need to get stuff like that, then we get stuff like that. There are a lot of different ideas that we can do. So please, you contribute these ideas. Let's work on this together. And I really can't wait to see what you come up with. Thank you so much. At this point, I'm out of surgery. Hopefully I'm recovering well. As soon as I can, I will email everyone and just going to let you know how I'm doing. Thank you. Bye-bye for now.